We're going to jump up to talk about photosynthesis at the leaf level. Here's some pictures of measurements being taken on a pine tree using a Lycor 6400 photosynthesis unit. Basically, you clamp these leaves into the chamber and measure the net exchange of CO2 and use that to infer net photosynthesis rates. When we measure net photosynthesis rates using an instrument like this, we usually express the units using the most common units are micromoles per meter squared per second, which basically is micromoles of CO2 per meter squared of leaf area per second. It's usually written, something like micromoles per meter squared per second. That's usually how those units are written. So it's expressing the net uptake of CO2 in micromoles. That's a number of molecules per meter squared normalized leaf area per second. Sometimes photosynthesis might be expressed on a mass basis. So you might see nanomoles per gram per hour, for instance, or something like that. And that's where this is nanomoles of CO2 per gram leaf tissue dry weight per hour, or something equivalent. So at any rate, it's some amount of CO2 taken up per some amount of leaf per unit time. Measurements of photosynthesis with gas exchange are almost always net photosynthesis. And it's important to understand when we see these numbers in the literature or when we're thinking about them that they are net photosynthesis. And what that means is that in reality, those measurements are incorporating two different physiological processes at least. They're incorporating gross photosynthesis, which is the uptake of CO2 within chloroplasts, which is photosynthesis that we've been talking about. So that's gross photosynthesis. But because we're measuring net uptake from the entire leaf, that also includes CO2 that's emitted from the leaf by mainly respiratory processes. So living cells in the leaf have mitochondria that in the process of respiration are releasing CO2 simultaneously with the uptake of CO2 by photosynthesis. So what we're measuring with these instruments is the net photosynthesis, which is the balance between gross photosynthesis, uptake of CO2, and loss of CO2 through respiration. So it's just important to remember that. That's usually not talked about in most studies of net photosynthesis. It's just implicit and people understand it. But we do need to remember that. And it'll become more explicit when I show you light response curves later in this lecture. When we talk about limitations to photosynthesis, one way we often partition those is stomatal limitations to photosynthesis and mesophyll limitations to photosynthesis. And to illustrate that, I'm going to use this diagram, which is just the cross-section of an angiosperm leaf, and I'm going to zoom in on this part of the leaf. So basically we'll be looking at a stoma along with a few spongy mesophyll cells. So the construct that we're looking at here is two different phases of limitation. This diagram, I think, does a nice job of demonstrating the limitations to photosynthesis. And they have this diagrammed as a series of resistances. So you can think of these limitations as resistance to the diffusion of a molecule of CO2 from the air outside a leaf all the way to the stroma of the chloroplast. So remember, the stroma of the chloroplast is where the dark reactions of photosynthesis occur. So all of these different resistances are barriers to movement of a CO2 molecule from the air outside to that point where it gets taken up by rubisco. And we can divide those limitations basically into two sets of limitations. These are stomatal or gas phase limitations, which are basically all of these. And that's basically limitations to CO2. If we had a CO2 molecule that was traveling from here outside in the air, through the guard cells, through the intercellular spaces, to the cell wall, that is all of these limitations right here. So that includes the boundary layer limitation. So that's limitation due to the layer of air along the outside of the leaf. The stomatal limitation, which is the limitation to that CO2 molecule diffusing through the guard cells. And then this is the intercellular space limitation. So basically that CO2 molecule has to diffuse through the intercellular space to the outside of the cell wall. So at that point then, we have another set of limitations that are broadly referred to as mesophyll limitations. So when we think about mesophyll limitations, that's all of these limitations. And it's basically, and we'd have to zoom in a lot closer, but those mesophyll limitations are basically the CO2 molecule moving through the cell wall. So this is the cell wall resistance. This is the plasma lemma, so that just means that's the plasma membrane, the diffusion of CO2 through the plasma membrane of this mesophyll cell. This is the cytosol. The CO2 molecule has to diffuse through the cytoplasm of this cell, and it has to reach a chloroplast, which is a plastid within the cell. So, and then it has to diffuse through the membranes of the chloroplast. So just envisioning this path, and then it needs to diffuse through the stroma to the point where it gets taken up by rubisco. A couple of things to think about is that these limitations associated with right here, these gas phase limitations, this is CO2 diffusing through gas. At this point, once it diffuses through the cell wall, the CO2 is diffusing through water. Now, CO2 diffuses through water many times slower than it does in air. But fortunately, these distances that we're talking about here are vanishingly small. So although CO2 diffuses very slowly through water, the distances are very small once we're talking about diffusion here. Basically, it's from the outside of the cell wall to a chloroplast, and it's just a distance of microns, very small distances, especially relative to, for instance, these distances. So those two sets of resistance then jointly are called stomatal or gas phase resistances and mesophyll resistances. And together, that's basically the set of resistances to uptake of CO2 by photosynthesis. Now, when I think about stomatal and mesophyll limitations to photosynthesis, to me, stomatal limitations are relatively intuitive. As the stomata close, that slows the diffusion of CO2 into the leaf and will lower the rate of net photosynthesis. That's relatively intuitive. Mesophyll limitations to net photosynthesis, on the other hand, are maybe a little bit less intuitive because they're related to a number of things, including structural attributes of the leaf, like the thickness of mesophyll cell walls, the distance, the size of the cells, and biochemical limitations. So the activity of rubisco can influence mesophyll resistance as well. So that's a little bit less intuitive. This graph, I think, does a decent job of showing how net photosynthesis, which is A, might change with different values of mesophyll conductance. So this is a little confusing because we were talking about resistances in the previous slide, and here we're talking about conductances. So one way to think about it is conductances is just the inverse 1 over mesophyll resistance. So a conductance is just the inverse of a resistance. So when conductances are high, or a large number, that means the resistance is small, and vice versa. So this shows how photosynthesis would change relative to leaf internal CO2 concentration in the intercellular spaces, which is what CI is, and how photosynthesis would change with different values of mesophyll conductance. When mesophyll conductance is infinite, which means there's no resistance to no mesophyll resistance, so basically this is 0, this becomes infinite. That's this line right here. So photosynthesis rate will be maximum at any rate of intercellular CO2. But as mesophyll conductance decreases, so this is as these values get smaller in this direction, the rate of net photosynthesis will decline because there's thicker cell walls, there's less rubisco activity, and so on. That causes the conductance, mesophyll conductance, to be smaller, or the mesophyll resistance or the mesophyll limitations to be larger. So mesophyll resistances, mesophyll limitations is just sort of a blanket term that describes these biochemical and structural limitations to net photosynthesis, as opposed to limitations to net photosynthesis caused by stomata, just the opening and closing of stomata. Carbon isotope discrimination is a widely used and really powerful method for looking at plant physiology. Isotopes of carbon can be used to infer variation in the ratio of photosynthesis to stomatoconductance. It's not immediately apparent why that's the case, so I'm going to spend a little bit of time explaining that. So to start with, need to remember that there's two predominant isotopes of carbon in the atmosphere in CO2. There's CO2 with a carbon with a molecular weight of 13, and that's about 1.1% of atmospheric CO2. Most of the CO2 in the atmosphere is the more common isotope of carbon, which is carbon-12. And that's about 98.9% of the CO2 in the atmosphere. But both of those are out there. And because of the molecular weights of those two isotopes of CO2, there's discrimination. Discrimination against this heavier isotope of CO2. And by discrimination, what I mean is when CO2 diffuses through the stomata, the carbon-12 version diffuses a little bit faster than the carbon-13 version. So what that means is that there is discrimination against carbon-13 in the stomata. In addition, there is also discrimination against the larger isotope of carbon in CO2 by rubisco. Rubisco preferentially interacts with the carbon-12 version versus the carbon-13 version of CO2. So there's a natural discrimination against carbon-13 in photosynthesis due to both that diffusion discrimination and that discrimination by rubisco. So by looking at differences in how much discrimination has occurred, we can infer differences in the physiology of the tree. So I'm going to give an example using a cartoon that I've drawn. So this cartoon is these boxes are leaves, and these leaves have a particular photosynthesis rate. So these circles of arrows inside are just representing that these two leaves are photosynthesizing at the same rate. But these two leaves have their stomata open differently. The stomata on this leaf are almost closed. So this leaf has lower stomatal conductance. Whereas this leaf, the stomata are wide open, so this leaf has very high stomatal conductance. The inverse, this leaf has a high stomatal resistance, this leaf has a low stomatal resistance. Okay, but their photosynthetic carbon uptake by rubisco is the same. So these two leaves differ in stomatal conductance, high stomatal conductance and low stomatal conductance. So what that means, they have the same photosynthesis rate, so the PS is the same in these two, but the ratio will be different. So because stomatal conductance is high in this leaf has a low ratio of photosynthesis to conductance, whereas this leaf has a high ratio of photosynthesis to conductance. So if we think about the leaf internal CO2 concentration relative to the CO2 concentration outside the leaf, because the stomata are wide open in this leaf will have a higher ratio of CI, internal CO2 concentration, to CI, ambient CO2 concentration, relative to this leaf. Because these stomata are closed, so the internal CO2 concentration of this leaf is relatively lower relative to ambient. Those differences in stomatal conductance will lead to differences in discrimination, because in this leaf, where basically the CO2 concentration is lower inside the leaf, what that means is that as photosynthesis takes up CO2, as it takes up carbon-12s preferentially, it basically will deplete the carbon-12s in these intercellular spaces and by chance start taking up more of those carbon-13. So I'm representing CO2 with the carbon-13 as these dark circles. So in this leaf will just end up taking up more carbon-13 because photosynthesis is depleting the carbon-12s. Whereas in this leaf, where there's ready diffusion of CO2 into the leaf, there will be higher discrimination against carbon-13. Whereas here there will be less discrimination against carbon-13 because the leaf will eventually take up more carbon-13 because there's less diffusion into the leaf. So this leaf will have less discrimination against carbon-13. So there's a measurement called delta. So we have big delta and little delta, which indicates the degree of carbon-13 discrimination that occurs. So this leaf will have higher big delta, which means more discrimination against carbon-13, or a more negative little delta, which also means more discrimination against carbon-13 than this leaf. So what that means, if we ground up these leaves and analyzed their amount of carbon-13 relative to the amount of carbon-12, we would basically find a relatively larger amount of carbon-13 in this leaf, either in sugars fixed by photosynthesis or in cellulose in that leaf, if on average, because on average it has a lower stomatoconductance. So what that means, these measurements of carbon isotope discrimination are an integrated or long-term measurement of the ratio of photosynthesis to conductance in these two leaves. So carbon isotope discrimination gives us a time-integrated estimate of the ratio of photosynthesis to conductance in a particular leaf, or if we're measuring in growth rings, for instance, that is sort of a plant-integrated estimate of average ratio of photosynthesis to conductance for that entire tree. So stable carbon isotopes are a really powerful tool for time-integrated measurements of the ratio of photosynthesis to conductance. So I'm going to explain in a little bit more detail what these measurements and how they're calculated on the next slide.